here today to share with you some of the results we have from our PRRS reproductive research. So we've conducted a number of animal trials over the last 5 years, and collectively they're known as the pregnant gilt models, and have a number of papers published. The experimental model we use is to inoculate a pregnant gilt at 85 days of gestation and to terminate somewhere between 12 and 21 days. But most of what I'll talk about today is termination at 21 days post-inoculation or 106 days of gestation. And during that period of inoculation, we collect blood from the gilts, and then at termination, we collect blood from the gilts as well as tissues and blood from the fetuses. And the blood and the tissues are used for a number of then ongoing analyses where we try to determine what the mechanisms of resistance are. So over the last 5 years, we have inoculated close to 200 or over 200 landrace gilts. Those landrace gilts are bred to Yorkshire sires, single sire basis, to produce a progeny, which is F1, which is obviously the commercial progeny that we use. We're using type 2 PRRS. It's a very virulent strain known as NVSL 7895. And we use reference controls, and in some of the work I'll show you, so it's a lot of work to terminate this many gilts, so we do it in different experimental replicates. And in each of those replicates, we have some reference controls. So this is some of the data from the first 115 pregnant gilts. The different colored dots represent the different experimental replicates, and you can see that there's no clustering by replicate. So we feel that makes the data fairly sound. And in this particular plot, what you're looking at is a 2x2 chart. On the horizontal axis, we have fetal mortality rate, and on the vertical axis, I'm showing levels of virus in fetal thymus. And obviously, for a fetus to die, they need to be infected, and the higher the levels of virus in the infected fetuses, the more likelihood they have of dying. And what's very interesting is the range of variations you see in both mortality rate and fetal thymus viral load. And in the top right-hand quadrant, you can see a very susceptible population of gilts. This is all gilt level data. So these animals have very high levels of mortality, above 50%, and they have high levels of viral load. And that's obviously a very undesirable characteristic. By contrast, in the lower left-hand quadrant here in red, you can see a population of fairly resistant animals. These have low fetal mortality rate, and these have low levels of virus load, and that's a very desirable characteristic. And in the top then left-hand quadrant, you have what we would call a tolerant set of animals, and these animals have low mortality rate, but you can see that their levels of viral load in thymus are fairly high. So in spite of infection and high levels of virus, those animals are able to tolerate infection. Now, at 21 days post-inoculation, the fetuses can be categorized into 4 different categories dependent on their status. So on the left-hand side in green, we have live fetuses. The viable fetuses on far left are those that look essentially normal at 106 days of gestation. They're white-skinned or pink-skinned. They would continue to grow, and they have very large and engorged umbilical vessels. Next to them are the meconium-stained fetuses, and those are live fetuses, but they're covered with fecal material, and we know that meconium staining is a sign of fetal stress. And these are fetuses that are typically very high viral load, and they are within a day or two of being dead fetuses. And then on the right-hand side, we have 2 categories of decomposed or of dead fetuses. Firstly, the decomposed, which have probably only been dead for a day. So they're largely white-skinned and have not decomposed that much. And then you see on the far right the autolyzed fetuses, and those are fetuses that have been dead for a number of days. They're in advanced stages of autolysis. And we use these 4 different categories to, in fact, reflect resistance. So on the far left-hand side, the viable fetuses are the most resistant in the litter. And on the far right-hand side, the odd-life fetuses are the most susceptible in the litter because those have been the first fetuses to die in the litter. Now, when you put those patterns together in situ and look at where they may be located within a litter, some very interesting patterns emerge. And what I'm showing here is data. Each row represents a litter, and each column represents a fetus and where they're located in the uterus. So on the extreme sides, R1 and L1 represent the farthest right and the farthest left fetus, which is closest to the ovarian tip. And then as the numbers increase, we move towards the body of the fetus, or body of the uterus. And remember that the left and the right fetus will be close to each other at the body as the numbers increased. So we see from the first row, we have a litter of control litter. So this is non-inoculated gilt, and all the fetuses are viable, represented by dark green. In the second row, we have a PRRS inoculated gilt, and in fact, all the fetuses are viable. So that's a very resistant animal where all the fetuses have survived up to day 21. The third row is another PRRS inoculated gilt, and that's another very resistant animal where we have 15 out of 18 fetuses that are viable, but we have 3 meconium-stained fetuses represented by the lighter shade of green. And those are the index fetuses in the litter, the first ones to start to see the effects of PRRS virus. And they are really located randomly throughout the litter. The fourth row represents a gilt, which is a very susceptible gilt. 94% of fetuses were dead in that litter, and the remaining live fetus was meconium-stained, so it's near death. The fifth row represents a litter that we have deads clustering in one horn, in the left horn, but we have all viable fetuses on the right horn, for whatever reason that is. And then the last row represents a gilt which has deads and viable that seem to be scattered throughout both the left and the right horns. So our overall overarching hypothesis, or goal in our research, is to increase PRRS reproductive resilience or resistance. And those are 2 different words, and they mean different things. So firstly, resistance means there's the ability of the host to control the pathogen life cycle. So what that means is there's something in the host genetics or the host phenotype that prevents virus replication. We don't know how that is, but that's it. Lower levels of virus, and that's a very important trait when controlling PRRS. By contrast, what resilience means is the ability of the animal to continue to perform even though it is infected. So productivity in the face of infection. And at this point in time, we're striving for both, either resistance or resilience. And as I progress in my research, I'm thinking that we're likely more to be focused on resistance than resistance. But for the sake of this talk, we'll say either would be satisfactory. Now, to achieve that, we have to look for different mechanisms. And some of those mechanisms are at play in the dam, or the mother. And those can be systemic events that are happening, such as genetics or immune response or her metabolism, or those could be at play specifically in the uterus or in the endometrium, and that can be what's happening at a cellular level, at a pathologic level, at a gene level. By contrast, we can also look at what's happening in the fetus, and that can be how the virus transmits across the placental tissue, what happens when the virus enters the placental tissue, something about the placental development of the fetus, or something about the fetus itself. And very importantly, it could also involve what's happening to adjacent or neighboring fetuses, because they have an impact as well. So we're going to start off by talking about events that are occurring in the dam. So after PRRS infection, virus replicates in tissues. It's circulating systemically in the blood, and then it continues to replicate as it goes into specific target organisms. So what we see here is a line plot of about 115 PRRS inoculated females, gilts. The horizontal axis shows the 4 different post-inoculation days, 0, 2, 6, and 21. And on the vertical axis, I'm showing levels of virus detected by PCR. And you can see that all gilts are negative at day 0, as would be expected. By day 2, they're 100% positive. Very importantly, look that at between day 2 and 6, the levels of virus continue to increase. And then by day 21, virus levels fall. And at day 21, post-inoculation, some of the females are negative. But for the most part, females continue to be positive. We know that PRRS is an immunosuppressive virus. And what we see systemically in the blood of the gilts after infection is a reduction in the number of white blood cells. And that is consistent across all the different types of white blood cells, from B cells to lymphocyte, or to T cells to T helper cells. And it occurs very, very rapidly. So what this plot is showing is, again, on the horizontal axis, the 4 different post-inoculation time points, 0, 2, 6, and 19. And in the y-axis, I'm showing numbers of white blood cells circulating in the blood of the female. And you can see with the white bars representing controls and the black bars representing inoculated gilts. And by day 2, we have a transient drop, it's quite a significant drop in white blood cells. And then we start to see a rebound by day 6. We still see lower levels in our inoculated gilts, but things return back to normal by day 21, or day 19 in this case. And that's across all the different subsets. PRRS is also known to be known to suppress interferon alpha levels. Interferon alpha is a cytokine that's very important for fighting viral infections. It's released very quickly, and it's one of those very fundamental cytokines that we rely on for our innate immune response to viruses. And when I looked at this chart initially for about the first 2 or 3 years, I was really confused by it, because I had been hearing all the way along that PRRS virus induces interferon alpha suppression. But what we see from this chart here is we have quite a spike and high levels of interferon alpha by day 2. This is measuring interferon alpha levels in gilt's blood. But what is not shown nearly as well on this particular chart is the suppressive effect of PRRS that occurs after day 2. And remember, we talked about virus levels in blood, and we saw an increase in virus from day 2 to day 6. And that, ironically, does not correspond with a continuing high level of interferon alpha, and that's the immunosuppressive effects of PRRS. We have an initial release from cells, immune cells, probably because it's preformed or it's before the impact of PRRS on those cells, but as soon as PRRS takes hold, it begins to replicate, it essentially shuts down interferon alpha production. Now, the suppression is better seen by this chart here. And again, on the horizontal axis, we're seeing our 4 different post-inoculation days. In this case, what we've done is we have collected white blood cells from dams after infection. So day 2 represents a blood collection point at day 2 after infection, and then day 6, and day 19. So those cells are circulating in the gilt's blood for a number of days and having the effects of PRRS on them. And then what we do is we take those cells back to the lab and we re-stimulate them with PRRS in 96-well plates, and we measure the amount of interferon alpha that they can in fact produce when they're re-stimulated. And what we see here is that by day 2, we start to see the effects, and then further by day 6 of the immunosuppression caused by PRRS. And that's the long-standing immunosuppression continuing out towards day 19. So we were interested in looking at what factors were potentially associated with low fetal mortality rate. And this is important because we're looking for different things that we could measure in gilt's blood and then use to select females in clean nucleus and multiplication herds before they're even exposed to PRRS. So that's the goal of what we're trying to do here. And there are a number of factors that are important that we could measure in blood. First would be the number of NK cells circulating. NK cells are effector cells of the innate immune system, so they exert a beneficial effect. Secondly, IL-10 is a cytokine that's involved in regulation of immune responses, and it exerts a beneficial effect. But surprisingly, interferon alpha, I've been promoting the effects of interferon alpha, but in fact, it's detrimental to fetuses. So what that means is that interferon alpha is a very important cytokine for fighting off viral infections in the dam, but the higher the levels of interferon alpha are really detrimental to the fetus. So we have this balance here that we have to control for. We've also spent some time looking at the transcriptome of the Gilt. So what this means is what genes are upregulated and downregulated in the Gilt after infection. And you can measure those in specific tissues, or you can measure those in blood. And in this chart, I'm showing what we're measuring in blood. And to do this is a very expensive technique, so you can only use a very small number of animals. We measured the transcriptome and compared it in the groups of susceptible animals in green to the number of resistant animals in red. The susceptible animals in green had a 76% fetal mortality rate and levels of virus in thymus above 4.4 log, where the resistant animals had less than a 5% mortality rate and lower levels of virus in fetal thymus. And what we found was that the low mortality rates, so those are the group in red, were more immunologically primed, and they had faster antiviral or immune responses, and both innate as well as adaptive immune responses. And there were 4 key sets of genes that were upregulated. First was platelets. Platelets are cells that are important for repair of vascular injury, obviously, but they also have an effector role in the innate immune system. Secondly, we saw an upregulation of genes associated with the inflammatory responses and interferon responses. And then thirdly, we saw an upregulation of the adaptive immune responses regarding T-cell activation as well as proliferation and signaling. So what this means is those are potential traits that we could select for if we had tools that we could actually measure those in Gilts prior to infection. So just to summarize some of the key messages related to systemic responses in Gilts. Firstly, interferon alpha levels appear to be detrimental to fetuses. More resistant Gilts appear to be immunologically primed and have faster adaptive immune responses following PRRS infection. And PRRS levels in sera, although they increase after infection, are unrelated to resilience. And I think what virus levels in serum really do is to seed the uterus with PRRS, which then crosses the placenta. So with that, let's then turn our attention to what's happening in the fetus and the placental unit. So just as a reminder, each fetus has its own placental unit, and the placenta has 3 different layers. The innermost layer is the amnion, and the 2 outer layers are the chorion and the allantois, and in the pig, those 2 layers are fused together to form the atlantochorion. And it's the atlantochorion which then is in apposition to the endometrium, and it connects through a number of interdigitating folds, which are seen here in this electron micrograph. So what we see here is a piece of maternal-fetal interface. There's like 2 pieces of bread that are put together with butter, and then they've been peeled apart separately. And the top half is the endometrium, and the bottom half is the placenta. And you can see all those big mountainous-like structures are the primary maternal folds, and then within each of those primary folds are a number of secondary folds. And the role of the primary and secondary folds are to increase vasculature so we have exchange of nutrients between the dam and the fetus. This is a micrograph of maternal-fetal interface, and you can see 2 pieces. On the left-hand side is the endometrium, and on the right-hand side of this image is the placenta. And both of those are coated with a layer of epithelium, which is represented by the dark purple nuclei. And the bar represents a piece of separation, or small piece of separation of the placenta from the endometrium. We know that PRRS causes apoptosis, and it's been shown for a long time to do that. And we were interested in knowing whether apoptosis was related to placental separation or was a cause of placental separation. So PRRS causes apoptosis of the macrophage it infects, and we call those CD163 positive macrophages. It kills them, but it also kills adjacent cells as well, including the uterine epithelium. We know that apoptosis is related to severity of PRRS, so higher viral load, higher, more severe vasculitis, and a greater odds of the fetus being meconium stained. But we were still unsure of whether placental separation is associated with apoptosis, and that'd be a very nice explanation for why fetuses die. Simply, the placenta separates because of apoptosis caused by PRRS, and everything goes away. All we have to do is stop that process, and PRRS would go away. Not quite so easy. So we've been investigating placental separation for a long time, and used a number of different experiments to do that. And in this particular plot, what I'm showing is placental separation, which we've just crudely graded as mild, moderate, and severe. And I looked at different fetal preservation statuses, and my hypothesis here was that if meconium staining is representative of clinical progression from viability, so we have viable fetuses, we have meconium stained, and then they die. I thought, well, there should be an increase in placental separation between viable fetuses and meconium stained fetuses, and what you can see from this chart, just looking at the black lines, it doesn't occur. So there's no progression in placental separation related to meconium stain. When we see placental separation increase is after the pigs die, after the fetuses die. But unfortunately, we don't yet know whether it's separating because the pig died, the fetus died, or whether it is separating and then the fetus dies. So that's still a mystery that we have yet to unfold. So a couple of key messages related to responses in the maternal-fetal interface. Firstly, we know that PRRS replicates in CD163 positive macrophages. We know that it induces apoptosis of those macrophages as well as the uterine epithelium. We know that the severity of apoptosis is associated with the severity of disease, but we don't yet know whether apoptosis is the cause of placental separation and the cause of fetal death. So there's ongoing work that needs to be done there. So what does cause fetal death? Well, there are a number of factors that we do know. The first factor associated with fetal death is interferon alpha levels, like we've previously talked about. Second factors are really what's happening in adjacent fetuses, in the neighbors. And if I'm a fetus and I have two fetuses on either side, the more fetuses that are dead or the more fetuses that are virus positive, the greater likelihood that I will be a dead fetus as well. Virus levels in both the fetus and the maternal-fetal interface are very important. All those factors are detrimental. And then there's one factor that has a beneficial effect, and that, again, is the number of T helper cells that are circulating in the Gilt's blood. Now, viral load is obviously a very important factor as well, or a very important phenotypic trait. We try to keep virus levels low in fetuses, and that'd be a desirable outcome. Again, there are a number of factors associated with viral load. Firstly, what's happening to neighboring fetuses. The higher viral load in those fetuses, they exert a positive impact, or I guess a negative impact on a given fetus. The levels of virus in the maternal-fetal interface is correlated with the level of virus in the fetal thymus. Fetal preservation status is very important. Meconium-stained fetuses have, in fact, the highest levels of virus, which are then followed by decomposed and autolyzed fetus. And then there are a couple of factors that have beneficial effect on fetal viral load, one being myeloid cells and the second being a cytokine IL-12, which is circulating in the blood of the dam. There's another very important factor related to viral load in fetuses, and this is something that we stumbled across, and it took us a long time to really understand and believe what we're seeing here. So what I'm showing here is a line chart, and the four different colors of lines represent viral load in fetal thymus. And for this particular analysis, we categorized the level as negative, low, medium, and high. And I want you to just to focus on the yellow line and the blue line for this example. On the x-axis, what I'm showing is the level of intrauterine growth retardation. We know that all fetuses don't grow the same. And sometimes placenta doesn't develop as well as other neighboring fetuses, so some of these fetuses become runts. So on the far right-hand side, I'm showing fetuses that have extreme intrauterine growth retardation. They are the prime runt pigs of the litter. On the left-hand side, I'm showing the opposite. So those are the fetuses that are large and fat and healthy, and we love those fetuses because they grow well the minute they hit the ground. The blue line is the probability of a fetus being negative, PRRS negative, at birth. And you can see that the probability is very high in runt fetuses. And by contrast, the yellow line is showing fetuses that have very high viral load, and the probability is very high in the large fat fetuses. Completely opposite to what we had expected. That I'd always thought the runt fetus would be more susceptible to disease, but in fact, the exact opposite is occurring. And we think this is because the large fetuses have a larger placenta, and there's more opportunity for virus transmission to those fetuses. Unfortunately, that doesn't mean that the solution to PRRS is to create a bunch of runt pigs, but it may in fact work, but obviously that's not something we want to go to. So what are some of the solutions to PRRS? Well, we'll move into some of the genomics work that we've done, and this work has been done by a grad student called Tian-Fu Yang at the University of Alberta. We have inoculated over 200 animals. We have over 1,700 fetuses that we have phenotypic data on. So it's a larger study and allowed us to do something called a genome-wide association study. And this is used to measure and identify different parts of the genome that may in fact be associated with resilience to PRRS. So the first trait that we looked at is fetal viral load. Again, that's a very important characteristic or phenotype of fetuses where we expect low viral load to be beneficial. And in this case, we're showing a Manhattan plot. And the Manhattan plot along the x-axis is showing the 19 chromosomes that are present in the genome of the pig. And each of those dots represents a single SNP or a single nucleotide polymorphism. And there's 3 that are circled in red, which show significant SNPs that are associated with viral load. And together, those SNPs explain about 13% of the variation in viral load. And there's one in chromosome 1, one on chromosome 12, and one on chromosome 14. And if we delve a little bit deeper into each of those SNPs, we can see the effect that each one has on viral load. So for each of these box plots, the y-axis is showing viral load in log 10, and it ranges from 1 to 5. And we have our 3 SNPs. First box is chromosome 1, second box chromosome 12, and third box is chromosome 14. And for each of these places on the chromosome, we have 2 alleles. One comes from our mom and one comes from our dad. And it can be A or it can be B. So some of these fetuses are AA, some are AB, and some are BB fetuses. And you can see the number of fetuses in each of those 3 different categories. So for the first and the third box plot here, so that's chromosome 1 and chromosome 14, you can see that virus load decreases as the number of A alleles increase. So A is the beneficial or the favorable allele, and the allelic effect is about half a log. So fetuses that have 2 A's compared to 2 B's have viral load, which is about 1 log lower, and that's pretty important. The box plot in the center is for chromosome 12, and that shows an allelic effect which is favorable for the B allele, but it's a recessive trait. So that means only the fetuses that have 2 B alleles, or BB, show the reduction in virus load in fetal thymus. Now, these are 3 different alleles, or three different places on the chromosome, and they're all acting independently. So we ask the question, what happens if there's a fetus that has 6 favorable alleles, and how do those compare to fetuses that have very few favorable alleles? And that's what this next plot shows. So this is a histogram. There's 3 separate plots. Each of them on the horizontal axis is showing the distribution of fetuses and the virus load that they have. And on the Y plot, it's showing the relative proportion of fetuses. The top plot is fetuses that only have 1 favorable allele. The middle plot is fetuses that have 3 favorable alleles. And the bottom plot, histogram, is fetuses that have 5 favorable alleles. And you can see that as you move from top to bottom, the proportion shifts from right to left. And the number or the proportion of fetuses that have no viral load or are completely negative increases from about 20% through up to over 60%. So clearly there's an advantage that the lucky fetus are the ones that have more favorable alleles. So we think this is a very important finding. Now, another phenotype we looked at was fetal viability. And this is a contrast between viable versus meconium-stained and decomposed fetuses. And for this contrast, we found one SNP on chromosome 7 that explained 35% of the variability in fetal viability. And that's a very important finding because that's a substantial genetic variance that we've discovered. And if we could find the candidate gene that is represented by that SNP and therefore select for that candidate gene, we can make a tremendous, we put tremendous selection pressure on resilience. So that's what our work is ongoing. So to summarize some of the key findings related to responses in the fetus. First, fetal death and viral load clusters and litters, and we think that may represent infection of the index fetus first and then lateral transmission within the uterus. We know that IUGR fetuses or runts are at greater risk of infection compared to the non-IUGR fetuses. So far, we have 4 single nucleotide polymorphisms that we potentially could use in the future to select for PRRS resilience. We have a lot of work to do between now and then, but hopefully we'll get there. And then finally, something we haven't talked about, and that is that lesions in fetuses are very rare, but when they occur, they certainly markedly increase the risk of fetal death. So now, this research has been briefly mentioned earlier this morning. This is work by Randy Prather's group at Missouri and many others, also now repeated at by the group in Edinburgh, which shows that if you knock out the CD163 receptor on the macrophage, that you can make pigs completely resistant to PRRS. And that is certainly very, very promising work. And as has been previously said, if this comes to fruition, maybe all of us will be out of a job that are studying PRRS, which is okay because I'll probably retire as well at that point. But I think there's a lot of steps between now and when this research will actually come to fruition. So hopefully it will. But in the meantime, our approach has been to help control reproductive PRRS by exploiting the natural variation in resilience that presently exists in our populations. In the last 5 years, we've really been focused in on the pathophysiology of reproductive PRRS to try to understand what's happening so that we can then be more intelligent in our approaches to control PRRS. And we have a lot of ongoing work that we will hopefully be able to measure items in blood, phenotypic or genotypic biomarkers, and use those to select for PRRS resilience in the future. And our goal ultimately is to really create tools to control PRRS that can be applied to any pig, any breed, and anywhere in the world. And with that, I'd like to again thank the organizers for the opportunity to speak with you today, as well as my collaborators and funders. Thank you very much.